Hello everyone, welcome to today's session. Our webinar this afternoon is an introduction of HLA Twin version 3.0, the new ABO kit, and analysis of interesting HLA cases. My name is Melista and I'm the head of product at Omixon. With me today is my colleague Libor Kolesár, who is one of our great field application scientists and principal presenter of this webinar. At the end of the presentation, there will be a live Q&A session where we will have the chance to answer any questions submitted during the webinar. Please feel free to type in your question at the question text box that appears on your screen anytime during the webinar. No need to wait until the end. Finally, a recorded version of this webinar will be available within a few days. All registrants will receive a link by email. If you have any feedback that you would like to share with us, do not hesitate to send it using the sales@omixon.com or marketing@omixon.com email addresses. We are ready to begin. We'd like to start with an agenda of today's session. The first item will be a short introduction of our product, followed by the new features of HLA Twin version 3.0. Subsequently, we will talk about the ABO genotyping kit and the ABO analysis using HLA Twin. Finally, we will be reviewing a few HLA special cases and demonstrate how HLA Twin may resolve them. We will end with a Q&A session where we will be answering as many questions as we can. As most of you know, Omixon's flagship product is Holotype HLA, but let me give a quick summary of it for the participants that are not as familiar with it. So Holotype HLA is an NGS-based assay for HLA genotyping on the Illumina platforms. The whole protocol requires about 3.5 hours of hands-on time from beginning to end, 1.5 hours of hands-on time if it's automated, and allows you to go from DNA to genotyping in less than 48 hours. The workflow can be broken down to 4 parts: the sample preparation part, the library preparation part, sequencing, and data analysis on HLA Twin, where our focus will be today. And on that note, Libor, can you please walk us through the new features of HLA Twin?
Thank you, Effie, for the nice introduction and for introducing me. So good afternoon, everybody, again. As you probably know from our website, we are going to release a new version of our Holotype kit, version 3. And for that purpose, the new HLA Twin 3.0 has been developed. But let me first briefly describe you the upcoming changes in the new Holotype HLA kit v3. We have changed the PCR reagents we use to achieve more robust amplification. We have also simplified the PCR preparation using ready-to-use master mixes. The master mixes will save you time and reduce the risk of use error. Based on your feedback, we have also lowered the amount of input DNA significantly. We have also adjusted and changed some steps in the amplicon processing, so we are now able to load more samples onto the currently used Illumina flow cells. You'll be able to load even 288 5-locus samples onto the standard 300-cycle flow cell. You don't need to be worried about the compatibility of HLA Twin 3.0 and our kits, because it supports all our Holotype product lines. Version 1, version 2, and version 3. So you need only one version of HLA Twin software. Analyzing v3 data is very easy. Simply go to application settings before the analysis and select the HLA v3.0 kit. Actually, you need to do this if you want to reanalyze your v1 or v2 kit-based data from the past. Unfortunately, it's not possible to automatically detect the used assay version. With the introduction of new products, ABO and MIC-A genotyping kits, we had to restructure the allele database, so I will explain to you why you need to install the updated IMGT database format if you upgrade from the previous HLA Twin versions. HLA Twin works with many different databases. The IMGT HLA database, allele frequencies database, and NMDT linkage disequilibrium database. With the implementation of ABO genotyping, we had to introduce also an ABO database. However, its structure and format is different than the original IMGT HLA database, which contains only HLA and MIC-A alleles. Thus, we had to restructure the IMGT and ABO databases to be able to merge them together and enable their analysis. After the upgrade from the previous HLA Twin versions, the originally installed IMGT HLA databases will contain in their names extension deprecated. And if you attempt to select them, you will receive an error message that the database format is not supported, just as you can see on the screenshot. Just remove the database and download it again. It will be downloaded in the new format and you'll be able to upload it and use it successfully. And what about your old result files? No worries, the data analyzed with the old database format is still fully supported. The next new feature is related to the statistical genotyping algorithm. Previously, the statistical genotyping algorithm was able to analyze exons only. From this HLA Twin version on, you'll be also able to analyze introns and UTRs with statistical genotyping. That means that you can achieve higher resolution with the SG algorithm, with results reaching even the fourth field. On the screenshot, you can see the comparison of statistical genotyping based results in version 2.5 and 3.0. You can see that now the resolution is 4-field result. You should keep in mind that the statistical genotyping is the algorithm that relies on the IMGT database and is limited to the known alleles. By default, the current analysis in HLA Twin triggers the SG only in cases where at least one of the QC metrics is flagged as yellow or red. On this slide, you can see screenshots from HLA Twin genome browser displaying the consensus sequences and their coverage depth. In the upper part here, taken from HLA Twin version 2.5, you can see the coverage of exons only. Here are the exons shown in blue sequentially to each other. Exon 2, exon 3, exon 4. While in the lower part, which was taken from HLA Twin 3.0, you can see the coverage of both exons and introns. Here, the blue shown exons are followed by the yellow introns. Although the reporting and type preserver is not a new feature, I would like to give you a little more explanation on that, as not all people working in the labs are IT specialists. This feature was already introduced in HLA Twin 2.5, however, it has been further improved in HLA Twin version 3.0. Simply imagine that the architecture of HLA Twin has been divided into the type server and reporting server. What is the purpose of those two units? The type server is physically doing the data analysis, while the reporting server is the part of the software that allows the user to access the analyzed data for review, approval, and reporting. With physical separation of these two processes, the computer can use its resources more efficiently, and HLA Twin reaches more stability and power. And you can then connect as many clients as you need and enjoy the power of networking. The supported operation system for the server is either Windows or Linux, and for client stations, the supported operation system can be commonly used Windows, Linux, or Mac operation system. Using the server client configuration is always the best solution for your laboratory, as you can use the automated data analysis. The MySeq machine will save the output data onto the server machine where HLA Twin is running. And once the data appears in the defined output folder, HLA Twin will automatically trigger the analysis. So when you come back to the lab in the morning, you can start assigning alleles immediately. Last but not least, the HLA Twin CE that contains all the features mentioned above is currently under the testing and will be released for public use very soon. As always, the CE-IVD users will be notified as soon as it becomes available.
Thank you, Libor, for presenting us with the new features of HLA Twin. As Libor mentioned, one of the new features is that ABO genotyping is also supported in HLA Twin version 3.0. Additionally, Omixon is pleased to introduce the ABO genotyping kit, or as we call it, the monotype ABO. The most significant advantage of ABO molecular typing is the ability to achieve allele-level typing results, in addition to the phenotypes as shown in the screenshot. In this example, you can see the O phenotype here, followed by the allele name right next to it. The ABO gene is located on the Q arm of chromosome 9 and is about 20 kb long. The ABO-specific primers in monotype ABO amplify from intron 2 to the 3' UTR, resulting in a 7 kb PCR product. Here is a picture displaying the exon and intron structure of the ABO gene. As you can see, exon 1 does not contain any polymorphisms that distinguish the three alleles, A, B, and O. Thus, it is not required for allele-level genotyping. The monotype ABO kit comes with pre-mixed master mix for amplification. All you need to do is add ABO-specific primers and DNA to get started. It also follows the same workflow as holotype HLA v3.0. This means that if you are a holotype HLA user, everyone is inherently trained in the protocol. Furthermore, monotype ABO uses the same reagents as holotype HLA, which means that the amount of reagent QC that is required in the lab is reduced. To make this approach even more cost-effective, you have the ability to combine ABO libraries and HLA libraries in the same sequencing run. Finally, we will soon offer the option to also pull ABO amplicons with HLA amplicons using only one index per sample, which reduces the amount of hands-on time and overall turnaround time for the lab. So how do I analyze ABO in HLA Twin?
With this HLA Twin release, you'll be able to analyze both HLA and ABO genes using the same piece of software. You have two options, actually. You can either click on the Advanced Genotyping button on the home screen and select the ABO gene under the targeted genes setting, or you can create your customized protocol for ABO gene analysis and make it more user-friendly. To do that, go to Application Settings and click on Analysis Protocols under the General section. Here, click on Create New Protocol. At the top, click on the tab called Targeting Configuration. Here you can see three sections: Targeted Gene Family, Targeted Gene Set, and Targeted Genes. In the Targeted Gene Family section, select ABO. The Targeted Gene Set will be grayed out as they are related to the HLA gene family only, and enable the user to select quickly predefined sets of genes based on the holotype HLA kit that you are using. So you may move directly to the Targeted Genes sections underneath and select the ABO gene. To save the protocol, click on Finish button. With this protocol, you will be able to analyze the ABO gene only. Now you can see that you have two protocols in HLA Twin, the factory default one and the ABO one. If you go back to the home screen now, select the FASTQ file of your ABO sample and click on the Advanced Genotyping button. On the first screen, select the ABO protocol and click on Finish. The analysis of the ABO gene will start immediately. So that was all from a technical point of view. And now let's move on a bit and review a few special cases of samples with challenging results and interpretation. We have prepared a selection of samples that our customers encountered during their routine work, and I would like to use them to show you some features and tricks in HLA Twin. This first sample is representing a case when you get the second field ambiguous result due to a phase break. As you are all aware, the advantage of next-generation sequencing over standard SBT is that next-generation sequencing can tell you whether the nucleotide at the very first polymorphic position is on the same chromosome like the latest polymorphic nucleotide. We call this phasing. In HLA Twin, there are 23 different quality control metrics, and one of them is called fully phased consensus, like you can see here in the screenshot. Here we have the result of DQB1 locus 06030101 and 06090101, which is ambiguous with allele pair 0641 and 0688. All 23 quality control metrics are passing with a green traffic light, except the fully phased consensus. Because this is a second field ambiguity, it's important to resolve it before reporting the result. So how can this be resolved? Fully phased consensus QC metric is referring to the so-called phasing track in the genome browser. So let's jump in the genome browser to inspect it. To go to the genome browser, you can select the DQB1 locus right here on the left. In the genotyping sample results screen, then select the G-level result, which will enable you to display both allele pairs or all 4 alleles in the genome browser. And finally, click on the Browse Alignment button. What you can see here are 2 consensus sequences of DQB1 locus. Consensus 1, consensus 2. The phasing track is highlighted in the orange rectangle. We can see we can see that there are two phased regions, phased region 1 and phased region 2. This is why HLA Twin is reporting no fully phased consensus. There is a break between the two phased regions. What does it mean that I have two phased regions? It means that these SNPs are on the same chromosome within region 1, and those SNPs are on the same chromosome within region 2. However, due to the phase break, I cannot conclude that this block of SNPs and this block of SNPs are on the same chromosome. When zooming into the phase break, we can see some numbers in the variance track here, straight and cross. What does it mean? Straight numbers mean that there are 3 reads having this T nucleotide on the same chromosome as this A nucleotide, and there are 4 reads having this deletion on the same chromosome as this G nucleotide. On the other hand, cross numbers mean that there are 0 reads having this A nucleotide on the same chromosome like this G nucleotide, and vice versa. There are 3 reads that support that this deletion is on the same chromosome like this A nucleotide. If you compare the numbers, straight versus cross, HLA Twin doesn't have enough data to distinguish between the 2 nucleotide configurations, thus the phasing is lost. This results in a cis-trans ambiguity. So let's come back to investigate our DQB1 result and answer why the result is ambiguous. We must compare both allele pairs in the genome browser. We can see that alleles 0641 and 0688 are not fully defined in the IMGT database, so we can use only exons to distinguish them from the other allele pair. If we first focus on exon 2, we can see that the nucleotide sequences of allele 06, 03, 01, 01 and 0641 are actually identical, and the same applies for the other 2 alleles 06, 09, 01, 01 and 06, 88. So there are no separating differences in exon 2. Looking at exon 3, there is only 1 SNP difference between all 4 alleles, this GA nucleotide. Due to the phase break and the limited amount of reads available, as shown on the variance track, it's not possible to conclude whether the exon 3 that contains the A nucleotide belongs to allele 1, and exon 3 that contains the G nucleotide belongs to allele 2, and vice versa. So to resolve the phasing issue, we should try to reanalyze the DQB1 locus with more reads. After the reanalysis with 8,000 reads, we get only 1 phased region. And the allele ambiguity has been resolved. HLA Twin is now calling only 1 allele pair, DQB1 06, 03, 01, 01 and DQB1 06, 09, 01, 01. So there is no cis-trans ambiguity at all right now. The next sample represents the novel allele. In this case, we got a result of HLA-A 33010101 and 6603 novel allele. The novel alleles are always tagged with a hashtag and number 1 plus a flag. All quality control metrics are passing and the overall results look very good. When a novel allele is detected, it is important to confirm the validity of the novelty. In order to do that, let's click on Show Novelties button. This brings you to another window showing you the newly detected polymorphisms. There are actually 3 new polymorphisms. 1 in exon 3 and 2 in intron 5. We are interested in the one located in exon 3, as this one may have an impact on the amino acid sequence in the conic region. Select the first row and click on the Jump to Novelty button to check the new SNP in the genome browser. You will be redirected immediately to the genome browser to the novel position. In the A6603 novel allele track, you can see 2 reference sequences. One that belongs to the new allele and one that belongs to the closest matching already known allele. As you can see, the new SNP is a T nucleotide in exon 3 of the A66 allele, while closest matching allele 6603 is expecting a G nucleotide at this position. Thus, we would like to know whether there is sufficient evidence for this new nucleotide. Let's open the base statistics window. In this table, you can see that there are 82 reads that support T nucleotide in the novel allele. And there are actually no reads that support any of the other nucleotides at this position. This confirms the T novel nucleotide. One of our favorite features in HLA Twin is the ability to check whether the new polymorphism has an impact on the amino acid sequence. Simply click on Display Amino Acid button to show the amino acid track, right here. You can see that this new polymorphism actually exchanges glutamic acid with a new stop codon. Isn't it great? This feature can help you make a decision on the next steps and how to report this sample. The third and the last example is the detection of contamination or the presence of a third allele. In this example, the majority of the quality control metrics for all loci are failing, and this is shown with the red traffic lights. Here. As you can see, among the loci, a lot of rare and novel alleles are reported, which is highly unlikely. Let's check what's happening with the QC metrics of this sample. The following primary QC metrics are failing across all loci: key exon spot noise ratio, consensus coverage key exon minimum depth, and the noise ratio. This is a typical example of mixed signals coming from a contaminated sample. To confirm, we should check the short read alignment and the noise in the genome browser. So let's use HLA-A as an example. What we see here is that the short reads are not overlapping to form a continuous consensus sequence. There are big gaps throughout the consensus, and also there are some isolated read blocks that are entirely dissociated from the rest of the sequence. This effect shows that the algorithms had a lot of trouble constructing accurate consensus sequences. At this point, we would like to check whether there are any reads that have been considered as noise and have been filtered out. We can see these in the noise track. To turn on the noise track in the genome browser, just click on the Display Noise button. In this example, the noise track looks very messy. We can clearly see that the reads that have been considered as noise are forming at least two consensus sequences that are different from the ones that are displayed in the genome browser. All the colored lines represent different signals. So this concludes the demonstration of special cases and how the samples can be resolved using HLA Twin.
So thank you very much, Libor, for that very thorough and clear explanation of samples. I think a lot of people found it very useful. I have already seen a few messages about that. So if you would like to find out more information on Omixon's products, complex case data sets for practice with guidelines just like the ones presented in this webinar by Libor, just feel free to visit the Omixon website or email us directly. Additionally, a recording of this webinar will be uploaded on the Omixon Academy page of the website within just a few days. So thank you all for your attention. We will be moving into the Q&A section now, answering as many questions as we can in the time that we have left. We are happy to receive even more questions in the meantime. So let's see. Let me just take a look very quickly through the list here. And the first one I see is, here we go. So there is one here, it says that I have two MySeq machines. Do I need two HLA Twin installations, or can I analyze my data with one HLA Twin installation? I think Libor can answer that for us.
Yeah. So I would first ask you whether you run both MySeq machines at the same time, or you are using the second machine as a backup machine. So if you are running both MySeq machines at the same time, then I would encourage you to use two different HLA Twin installations, actually for each MySeq separately. In this way, you can speed up the data analysis. Otherwise, HLA Twin will process first data from the earlier finished MySeq run and then from the second MySeq. So if you use the second MySeq only as a backup, there is no need to have two HLA Twin installations.
All right, thank you, Libor. Another one that I see here is, when will holotype HLA v3 be available? And I can answer that actually really quickly. So holotype HLA v3 is under external testing right now, and it will be publicly available within the next couple of months. By the end of October, it should be expected. So another server-related one. It looks like there are a lot of server-related ones. Will my lab need both a typing and reporter server? That's actually a very commonly asked question.
Yeah. I have heard this question a couple of times before. I think that you are asking whether you need two physical machines, one that is dedicated to typing server and one that is dedicated to the reporter server. So actually, the answer is no, you don't need two physical machines. You just need actually one machine, yeah? Because the typing and reporter server actually shares the same resources of the server machine. So the difference is that the reporting server will use more RAM memory and more computer resources because it does all the calculations, while while the reporting server is just to let the user access the processes for approving results and exporting the results. So it doesn't need a lot of computer resources.
Thank you, Libor. Another fairly relevant one, software-related one. What limb system? Oh yeah, what limb system is HLA Twin v3.0 compatible with? That's another common one. So Libor?
Yeah. So actually, the HLA Twin cannot be directly connected with the limb software. HLA Twin instead is exporting the results to different formats. So the very commonly used HML format, which is quite similar to XML format, but a little bit different one, or the JSON format, or for user-friendly into the user-friendly format, the PDF. So the answer is actually the HML format, and these files can be then imported to your limbs.
Thank you, Libor. I have another one here. It says, how often is the database for HLA typing updated from IMGT to Twin? Do you want to answer it?
Yeah, I can answer that. So once the IMG database is officially released, our users will receive within 6 months the updated HLA database, and they will be automatically updated or informed by the software with message saying there is a new IMG database available. Would you like to download it? Yeah.
So another one here, also software related, how this new statistical genotype deals with alleles without reference analysis is impacted by alleles without no introns. So the answer to this is essentially, even though the new statistical algorithm allows for analysis of intronic regions as well, exactly because it relies on the IMGT database and any information that exists in that, if the intronic areas are not characterized in the IMGT database, then the statistical algorithm will not report them either. So it will be the same as if doing exons analysis only. Yep.
Here is a question related to a new product. So which reagents are the pre-mixed master mix that you mentioned? So in our kit, you will find the PCR components already premixed, including buffer, DNTP, Taq polymerase. And you will just need to combine this pre-master mix with the primer pair, DNA, and water. So that's it. And it will significantly reduce the time that you spend for the PCR preparation, and also it will reduce the risk of user error.
Thank you, Libor. There is another one here that is probably relevant. Do I need to purchase additional equipment or reagents to run this ABO NGS kit? And I guess the answer varies whether you are a current holotype HLA user or not. If you are a current holotype HLA user, then you don't need anything that you don't have in your lab. You already have absolutely everything that you need. If you are not a holotype HLA user, you need either a plate fluorometer or a qPCR machine, either one, and the sequencer, really. That's all. Yeah.
So there is one commonly asked question. Yeah. So the question is here that my MySec machine is storing data on the dedicated storage for storing data only. So I can't install HLA Twin software, actually no third-party software at all on it. So can I still use the automated data analysis? Well, yes, of course you can. The only thing you need is the server where you can install HLA Twin software and which is able to see the data stored on the data server. By default, HLA Twin is saving result files into the same location like FASTQ data. So first, check whether you have enough rights to write data on the data server. Yeah, and if not, you can bypass that by setting HLA Twin to store your results into a different directory than FASTQ data.
All right. Yeah, that's definitely a common one.
Yeah.
For sure. So another question is, let's see, can I use also the miniseq sequencer, the miniseq for sequencing for both holotype and ABO? I assume the question is.
Yeah, Effie, definitely you can. Our products work very well both with miniseq or MySeq or NextSeq machine. So definitely the answer is yes. And we have a couple of customers using the miniseq machine also under the accreditation.
Thank you. So just as we were asking the miniseq question, we were answering, sorry, the miniseq question, another interesting question just came in. Is this product compatible with the Illumina iSeq 100 instrument? Excellent question. So we are actually just starting to test the iSeq instrument with our products. The initial information seemed like that is definitely the case, but we will be investigating this a bit more, and we will be making an announcement on that later on. But that's a great question.
Yeah, that's good. That's a good one.
Mm-hmm. So there is another question here that is relevant to the analysis of HLA and ABO. It says, how long will it take for HLA Twin to analyze an HLA library combined with an ABO library? And the answer to that, I can answer that. So the answer to that is that essentially there are two separate analysis pipelines. So there is a pipeline for HLA data analysis and a pipeline for ABO analysis in the software. So each one of them are analyzed separately. So the analysis time actually is not affected.
And I can see one question. We have a data integrity issue. So do we need a server to connect to you for support? Well, actually, we don't need to access your server. If you can connect us with some application like Skype or GoToMeeting, WebEx to see actually what you are doing online, it's more than welcome. And if not, we can support you, for example, on phone, or if needed, we can even come on site to give you a troubleshooting.
All right.
Yeah.
Thank you, Libor. I guess we should address one last question that just came in as well. I'm interested in trying the monotype ABO. Well, thank you very much. What do I need to do for that? All you have to do is just contact your sales representative, or email us at sales@omixon.com, and we will provide everything that you need for that. Thanks for your interest. Okay, so I think this might be the last one, the last question. Thank you very much, everyone, for your attention. If you have any more questions or any feedback that you would like to share with us, as I said, feel free to email us. And looking forward to seeing you at a future webinar. Thank you.
Thank you.
Have a great day, everyone.
Bye-bye.